What if all of the world's biggest problems, from climate change to curing diseases to disposal of plastic waste, what if they all had the same solution? A solution so tiny it would be invisible. I'm inclined to believe this is possible thanks to a recent breakthrough that solved one of the biggest problems of the last century: how to determine the structure of a protein.
It's been described to me as equivalent to Fermat's last theorem, but for biology.
Over 6 decades, tens of thousands of biologists painstakingly worked at the structure of 150,000 proteins. Then, in just a few years, a team of around 15 determined the structure of 200 million. That's basically every protein known to exist in nature. So how did they do it? And why does this have the potential to solve problems way outside the realm of biology? A protein starts simply as a string of amino acids. Each amino acid has a carbon atom at the center, then on one side is an amine group, and on the other side is a carboxyl group. And the last thing it's bonded to could be one of 20 different side chains, and which one determines which of the 20 different amino acids this molecule is. The amine group from one amino acid can react with the carboxyl group of another to form a peptide bond. So a series of amino acids can bond to form a string, and pushing and pulling between countless molecules, electrostatic forces, hydrogen bonds, solvent interactions can cause this string to coil up and fold onto itself. This ultimately determines the 3D structure of the protein. And this shape is the thing that really matters about the protein. It's built for a specific purpose, like how hemoglobin has the perfect binding site to carry around oxygen in your blood.
These are machines. They need to be in their correct orientation in order to work together to move, for example, the proteins in your muscles. They change their shape a little bit in order to pull and contract.
But it would take people a long time to get the structure of just one protein.
Absolutely. So what should proteins look like was only started to answer really with experimental techniques.
The first way protein structure was determined was by creating a crystal out of that protein. This was then exposed to X-rays to get a diffraction pattern. And then scientists would work backwards to try to figure out what shape of molecules would create such a pattern. It took British biochemist John Kendrew 12 years to get the first protein structure. His target was an oxygen-storing protein called myoglobin, an important protein in our hearts. He first tried a horse heart, but this produced rather small crystals because it didn't have enough myoglobin. He knew diving mammals would have lots of myoglobin in their muscles since they're the best at conserving oxygen, so he obtained a huge chunk of whale meat from Peru. This finally gave Kendrew large enough crystals to create an X-ray diffraction image.
And when it came out, it looked really weird. People expected something kind of logical, mathematical, understandable, and it almost looked, I wouldn't say ugly, but intricate and complex, and kind of like if you see a rocket motor, right, and all the parts hanging off.
This structure, which has been called Turd of the Century, won Kendrew the 1962 Nobel Prize in Chemistry. Over the next 2 decades, only around 100 more structures were resolved. Even today, protein crystallization remains a big challenge.
Frankly, it is not uncommon that just a couple protein structures can be someone's entire PhD. Sometimes just one, sometimes even just progress toward one.
And it's expensive. X-ray crystallography can cost tens of thousands of dollars per protein. So scientists sought another way to work out protein structure. It only costs around $100 to find a protein sequence of amino acids. So if you could use this to figure out how the protein would fold, that would save a lot of time, effort, and money. What AI has done for proteins is just a hint of what it can do in other fields and on larger scales. In materials science, for example, Deepmind's GNOME program has found 2.2 million new crystals, including over 400,000 stable materials that could power future technologies, from superconductors to batteries. AI is creating transformative leaps in science by helping to solve some of the fundamental problems that have blocked human progress.
If you think of the whole tree of knowledge, there are certain problems where, if they're root no problems, if you unlock them, if you discover a solution to them, it would unlock a whole new branch or avenue of discovery.
And with this, AI is pushing forward the boundaries of human knowledge at a rate never seen before.
Speed-ups of 2x are nice. They're great. We love them. Speed-ups of 100,000 times, change what you do. You do fundamentally different stuff. And you start to rebuild your science around the things that got easy.
And that's what I'm excited about. These discoveries represent real step function changes in science. Even if AI doesn't advance beyond where it is today, we will be reaping the benefits of these breakthroughs for decades. And assuming AI does continue to develop, well, it will open up opportunities that were previously thought impossible, whether that's curing all diseases, creating novel materials, or restoring the environment to a pristine state. This sounds like an amazing future, as long as the AI doesn't take over and destroy us all first.